In the last few years, we've started doing more work looking at some of the cancer cell behaviors and trying to understand at the cell level and molecular level what's happening inside those cancer cells. We have three main areas that we're interested in, and that's breast cancer, prostate cancer, and bone cancer.
This is breast cancer, early stage breast cancer, the MCF7 cell line, and they look beautiful and healthy, so that's good. As healthy as cancer can be, right? Basically what I'm doing right now is I am taking all of the cells from these 6 plates and I'm moving them to 20 plates, so that way they can grow quicker and be happier when they have more space. And this allows us to have a lot of plates to do experiments on when we treat them with certain things to see how they react. So this is the very beginning of all of the steps in the lab.
Students here at George Fox, in particular in the sciences, are in a position to really do cutting-edge research, hands-on work that, in my experience, is the kind of things that are normally reserved for graduate students or for postdocs. The kinds of questions that they're asking, the kinds of hypotheses that they're pursuing are, I think, really complicated, really fascinating, and go way beyond the scope of your traditional undergraduate teaching lab.
This step is called running a gel, so we separate the proteins by size and their molecular weight. This gives us a way to crack open that cell and really look at what's going on and what's being affected in the cell, which allows us to understand how cancer is working. The best way to treat cancer is by knowing how it's different from our normal cells, because if we treat it like a normal cell, then all our normal cells are going to be affected too. So doing this allows us to look at different pathways, different enzymes that are affected, and then target those in future therapies.
You don't get results every single day, or it's really frustrating when you don't get results, but it puts it into perspective that everything that we're doing here is really helping the research go along, and that can help so many people across the world. It just made me want to come to work and work even harder in the lab and try to do as many experiments as possible over the summer, just to learn as much as we can.
Right now, I'm probing membranes. When we run an experiment, all the proteins that we collect end up on a membrane, and this Death Star, or this machine, it uses the photons to read kind of what the proteins are, and then they'll come out as these bands that show us what proteins are present, and that kind of tells us what things are active, what things are there that were in the cells, and then we can base our experiments off that. And so right now I'm scanning my membrane.
And why do you call it Death Star?
Because John likes Star Wars, so he calls it the Death Star. Not exactly sure if there's any more reason to that than that, but.
The work that we do in my research lab here, we study human cancer cells and use human cancer cells that other scientists and clinicians study in different places in the world. So our hope is that we can see things or discover things about those cancers that would be accessible to other scientists and clinicians. I think the students who do this kind of work get to learn a lot about themselves and how God has made them. How do they solve problems? How do they think critically? How do they work as a team? How do they work individually? And those are really important parts of the research experience, but it's also part of, I think, the mission here at George Fox, which is to help students grow in all facets of their life. It's really a great integration of trying to help students grow in their research and scientific expertise, and also grow as individuals.